Now we're going to look at the two classes of nephrons. There are cortical nephrons and juxtamedullary nephrons. 85% of nephrons are cortical nephrons, and they're almost entirely in the cortex. Just a little bit of their nephron loop dips into the medulla. Juxtamedullary nephrons are a lot closer to the cortex-medulla junction, and they have long nephron loops that really deeply invade the medulla. Their ascending limbs have thick and thin segments, and they're the most important in production of concentrated urine. So that's what we can see here. We can see some differences between cortical nephrons and juxtamedullary nephrons. And there is a short nephron loop for cortical nephrons, a long nephron loop for juxtamedullary nephrons, and that we see over here that the juxtamedullary nephron is a lot closer to the cortex-medulla junction, and its loop invades the medulla. Also, we see its second capillary bed is the vasa recta, where over here the second capillary bed for a cortical nephron is the proximal convoluted tubule. So for a cortical nephron, the efferent arteriole supplies paratubular capillaries, whereas in a juxtamedullary nephron, the efferent arteriole supplies a vasa recta. So if we look at the renal tubules associated with the two capillary beds, we see that the glomerulus in both produces filtrate, whereas in the cortical nephrons, it's the paratubular capillaries that reclaim the filtrate, and in the juxtamedullary nephrons, it's the vasa recta that reclaim the filtrate. Now, the glomerulus, we know, is the location of filtration because the adjacent podocytes of the visceral layer of the Bowman's capsule and the endothelial cells that are fenestrated of the glomerulus, when they come together, they create the filtration membrane. So that glomerulus with its filtrations is specialized for filtering. And that's different from the other capillary beds, so that's different from the paratubular or vasa recta. The glomerulus also is going to be fed and drained by an arteriole, and that's different compared to other capillary beds. We also don't see that for the paratubular or vasa recta. So the afferent arteriole brings blood to the glomerulus, and the efferent arteriole drains blood from the glomerulus. So afferent for blood arriving at the glomerulus, and efferent for blood exiting or leaving or draining from the glomerulus, however you want to think about it. Now, blood pressure in the glomerulus is high. The reason it's high is because the efferent arteriole has a smaller diameter than the afferent arteriole. So a lot of blood will flow into the glomerulus, and that blood will drain a lot slower. So we have a high blood pressure in that first capillary bed. Now, not as high as we see in the elastic arteries near the aorta, because remember, capillaries are definitely still fragile, and those fragile capillaries can be damaged by too high of a blood pressure. But the arterioles themselves are high resistance vessels, and they're able to dilate or constrict in order to control not only net filtration pressure, but glomerular filtration rate. Whereas the paratubular capillaries are low pressure. They're porous, and they're going to be adapted for absorption. And so that's why we saw especially those paratubular capillaries wrapping around the proximal convoluted tubule of cortical nephrons. They're going to be functioning there in reabsorption of water and solutes. They do arise from the efferent arterioles and cling very well to the renal tubule itself. And they empty into venules, specifically the cortical radiate vein. The vasa recta are a little different. They're long, they're thin-walled, and they're going to be parallel, kind of like in a network, to the nephron loops of juxtamedullary nephrons. They do arise also from the efferent arteriole, but only of the efferent arterioles that serve juxtamedullary nephrons. So we're looking here at a different capillary bed for juxtamedullary nephrons than that of the paratubular capillaries around cortical nephrons. And the reason we have these two types of nephrons, these differences, is because one of the nephrons, the juxtamedullary nephron, is going to function in the concentration of urine, whereas the cortical nephron is mainly going to be functioning as a reabsorption filter for solutes and water. So just to review, cortical nephrons are almost entirely in the cortex. Juxtamedullary nephrons have their long nephron loop almost completely in the medulla. We have paratubular capillaries for cortical nephrons, and we see the vasa recta for the nephron loop of juxtamedullary nephrons. So efferent arterioles are supplying different capillary beds depending on the type of nephron we're looking at. Next, we're going to look at the juxtaglomerular complex, or the JGC. Now, there is one JGC per nephron, and it involves modified portions of the ascending limb of the nephron loop, and the afferent arteriole. Now, it can also be the efferent arteriole. Now, the afferent arteriole and parts of the ascending limb of the nephron loop create, via some connecting cells, this JGC. And it's important in regulating the rate of filtrate formation and controlling systemic blood pressure. So make sure you know its main function. It has 3 cell populations: the macula densa cells, which are part of the distal convoluted tubule and sometimes part of the ascending limb of the nephron loop, and the juxtaglomerular cells, also known as granular cells. They secrete renin, and then extraglomerular mesangial cells. The macula densa cells are tall, they're closely packed, they're either a part of the distal convoluted tubule or parts of the ascending limb, and they contain chemoreceptors. And these chemoreceptors are going to watch the filtrate and see how much sodium chloride is in it. So they're regulating or sensing sodium chloride content of the filtrate that's entering the DCT. So that's why we find part of them kind of in that ascending limb and the other part in the DCT. The extraglomerular mesangial cells are between the macula densa cells. The macula densa cells and the granular cells. So the extraglomerular mesangial cells are located between the arterial and the tubule cells, and they're interconnected with gap junctions, and so they can pass signals between these two cell types. So let's go back one, and now let's look at the granular cells. The granular cells, also known as juxtaglomerular cells or JG cells, are found in the afferent arteriole. They can be in the efferent arteriole, but primarily afferent. Now, they are smooth muscle cells. They're enlarged. They are going to secrete renin, and therefore they're also going to use mechanoreceptors. They're going to be monitoring how much pressure is in the afferent arteriole, and then the secretion of renin can produce vasoconstriction, or if there is no renin being produced, it can cause vasodilation of that afferent arteriole and regulate glomerular filtration. So again, we have 3 cell types or 3 cell populations. The macula densa cells are located in either the ascending limb of the nephron loop or the first part of the distal convoluted tubule. The extraglomerular cells connect the macula densa cells to the granular cell population, and the granular cell population is also known as the juxtaglomerular cells or JG cells. The granular cells are located in the afferent arteriole, so they're kind of enlarged smooth muscle cells. And we're going to come back to the JGC. That won't be all you see of it, but let's just look at some kidney physiology first. So some basic mechanisms of urine formation. First and foremost, 180 liters of fluid is processed daily through the kidneys, but only about 1.5 liters of that ends up as urine. And there's 3 processes that are used to process 180 liters, and that is glomerular filtration, tubular reabsorption, and tubular secretion. So again, those are the 3 processes in urine formation, and they're adjusting blood composition as well. Glomerular filtration occurs in the renal corpuscle via the filtration membrane. Tubular reabsorption occurs in the renal tubule system, mainly in the proximal convoluted tubule, but can occur in the collecting ducts and distal convoluted tubule. Usually in the distal convoluted tubule, tubular reabsorption is hormone regulated, such as by ADH or aldosterone. Then tubular secretion occurs in the renal tubule system as well, but tubular secretion is primarily going to take place in the distal convoluted tubule and the collecting duct. So we do see some secretion in the proximal convoluted tubule, but mainly for really only the secretion of drugs, so the breakdown products of drugs. Glomerular filtration produces cell and protein-free filtrate. So this is where we filter blood plasma, and what can get through the fenestrations of the glomerulus and the filtration slits of the filtration membrane is able to move into the capsular space or the hollow space of the parietal and visceral layer of Bowman's capsule, also known as the glomerular capsule. Tubular reabsorption is going to be a selective process, and since glomerular filtration is not, it's able to select what it wants from the filtrate, which is in the tube, and return that back to the blood. So tubular reabsorption is a selective process that returns 99% of substances from the filtrate in the tube to the blood, specifically either the vasorecta or paratubular capillaries. Tubular secretion is the opposite of tubular reabsorption. So tubular secretion is also selective, but it's going to move substances from the blood back to the renal tubule system or back to the filtrate. You can also move these not back just to the renal tubule system, but to the collecting duct. So the kidneys are able to filter a lot of fluid, a lot of blood. They can filter the body's entire blood plasma volume 60 times each day. So they need a lot of oxygen in order to filter all this blood. They use a lot of active transport to both reabsorb and secrete these substances back and forth. So 20 to 25% of the oxygen that our body uses at rest is actually sent to the kidneys. And that's so that we can produce urine from filtrate. So filtrate is blood plasma minus proteins. It's the fluid that ends up in the renal tubule system from the capsular space, and it's produced by glomerular filtration. Now, from what we have in the renal tubule, which is part of the nephron that we're calling filtrate, what becomes urine is actually less than 1% of the original filtrate that flows from the capsular space into the proximal convoluted tubule. We're going to reclaim most of the substances that originally become filtrate in that capsular space throughout the rest of the renal tubule system, including the proximal convoluted tubule, the loop, and the distal convoluted tubule. What's mainly in urine is going to be water, and then metabolic waste and any unneeded substances, or ions in excess, for example. Here is a simplification or a schematic of a nephron. What we are also seeing is a simplification of the microscopic vascularization that occurs near the renal corpuscle. So we have a cortical radiate vein with an afferent arteriole leading to a glomerulus and then an efferent arteriole. They have the efferent arteriole merging into the second capillary bed, which here is a paratubular capillary, so we must be looking at a cortical nephron. Now, this part of the picture is representing the renal tubule system. So it's a stand-in, just this short tube for the PCT, loop, and DCT. And then we have a simplified renal corpuscle here with the glomerulus and capsule. What we're seeing occur at step 1 is glomerular filtration. So we're having fluid move from the fenestrated capillaries into the capsular space. Now remember, this is going to be large protein and cell-free, but other nutrients that fit through the filtration membrane get to move into the renal tubule system. Then we're going to have step 2, or tubular reabsorption, reclaim most nutrients. And then any waste products or breakdown products of drugs will move into the renal tubule system via tubular secretion. So this is a good image to use in order to study the 3 major renal processes that adjust plasma concentration of blood as we first filter, then reabsorb, and then secrete it in order to create urine. So again, glomerular filtration is a passive process, so no energy is used. We use hydrostatic pressure or a pushing pressure inside the glomerulus, the capillaries itself, in order to force fluid from the glomerulus into the capsular space. Now, small solutes and substances will move through the fenestrations and filtration slits of the filtration membrane, but large proteins and cells will not. There is no reabsorption that occurs in the glomerulus. So the filtration membrane again includes the porous fenestrations of the glomerulus itself, as well as the filtration slits created by the foot processes or the podocytes of the visceral layer of the capsule. So things like water, solutes, anything smaller than plasma proteins, even glucose, are going to pass through this filtration membrane. Here is a list of the 3 parts of the filtration membrane. We have the fenestrated endothelium of the glomerular capillaries, which is the endothelium of the porous glomerulus, the fused basement membrane that's shared by the fenestrated endothelium, and then last but not least, the foot processes of the podocytes. Podocytes have projections that line up on the basement membrane that is shared by the fenestrated endothelium and these podocytes, and it creates filtration slits or spaces where macromolecules are repelled, but small things like solute, sodium, potassium, and even glucose can pass as long as they fit through both the fenestrations and through the filtration slits. So that's what we're seeing here is the filtration membrane. The filtration membrane is located within the renal corpuscle. It's made by the adjacent structures of the glomerulus and the podocytes, or the visceral layer of the Bowman's capsule or glomerular capsule meeting. You can see the many projections of the podocytes, so there's slits or spaces in between them, and we can see pores of the fenestrated glomerulus. So if stuff fits through the pore and then it fits through the filtration slit, it gets to move past the filtration membrane into the capsular space of the glomerular capsule. From the glomerular capsule, then that filtrate will move into the proximal convoluted tubule. Here is a histograph of the filtration membrane. It is artificially colored, but you can see the filtration slits between the podocyte foot processes. So they are actually quite small. They're not even as large as indicated in that last image. Here is a kind of side or lateral view of the filtration membrane. It's simplified. We can kind of call it the clip art version. So here's inside the glomerulus, which is a capillary bed. We have plasma as our extracellular fluid. Plasma, anything that's in plasma can flow through the fenestrations, and if it gets stuck, it can't get into the capsular space. Things that are in plasma that are small enough to get through the fenestration and the filtration slits is able to move into the capsular space. So cells and proteins include plasma proteins, should never be in the capsular space. They will stay in the glomerulus. So macromolecules like plasma proteins and really large carbohydrates, other macromolecules, can get stuck in the filtration membrane. They are engulfed then by glomerular mesangial cells, and we'll see that only things that are small, so 3 nanometers about, can pass through the filtration membrane completely. So here's a quick list: water, glucose, amino acids, and nitrogenous waste. An example of a nitrogenous waste is urea. So those plasma proteins remain in the glomerulus, but they're used to maintain colloid oncotic or osmotic pressure, which is going to be a pressure that is different than the hydrostatic pressure. So it's a pulling pressure, whereas hydrostatic pressure was a pushing pressure. This colloid osmotic pressure that is maintained by plasma proteins like albumin is going to be able to prevent the loss of all water from the capsular space or all water from the glomerular capillaries. And then proteins in the filtrate would be an indicator of some type of problem with your filtration membrane, because those plasma proteins in general, should never leave the glomerulus or end up in your capsular space. So again, we have a couple of different pressures we were talking about. We have outward pressures that promote filtrate formation. So we have pressures that push fluid out of the capillaries and into the capsular space. The primary one there being the hydrostatic pressure that's in the glomerular capillaries itself. This could also just be called glomerular blood pressure. And this pressure in the glomerulus is the chief force pushing fluid out of the glomerulus, through the fenestrations, through the filtration slits, and then into the capsular space, or just out of the blood. And this hydrostatic pressure is pretty high for a capillary. It's about 55 millimeters of mercury. Most capillary beds are only about 26 millimeters of mercury for the pressure that's within their capillaries. But this makes sense. We were talking about the structure of blood flow going into the renal corpuscle. That afferent arteriole is a high pressure vessel, allowing the glomerulus to be a high pressure capillary bed. And the efferent arteriole is narrower, and it creates that high pressure dynamic in the glomerulus itself. So because the efferent arteriole is high resistance vessel with a diameter that's smaller than the afferent vessel, we create this quite high 55 millimeters of mercury hydrostatic pressure in the glomerulus. There's also some opposing pressures. There's inward forces. We have that colloid oncotic or osmotic pressure that would pull or keep the plasma proteins in the blood, but also pull or act on water to make sure we don't lose all water from the glomerular capillaries. Then we have that hydrostatic pressure that's in the capsular space. So there's a little bit of pressure in the capsular space itself, pushing back on the filtration membrane. But they're going to be much lower than the hydrostatic pressure of the glomerulus. So when we sum up all these forces, our net filtration pressure, or our result of summing up all these forces that are affecting filtration, is actually going to be an outward force of about 10 millimeters of mercury, or a pressure reading. That means that fluid's going to flow from the glomerulus to the capsular space through the filtration membrane. This pressure, which is responsible for filtrate formation, so the pressure that pushes things that fit through the filtration membrane into the capsular space, that is called the net filtration pressure. And it's the main controllable factor that determines glomerular filtration rate. It's actually directly related to glomerular filtration rate. If net filtration pressure increases, so does glomerular filtration rate. So here we can see all the forces that determine net filtration pressure. The main one's going to be that hydrostatic pressure in the glomerular capillaries. We have some opposing forces, that main one being the oncotic or osmotic pressure of the glomerular capillaries, but they can't beat the high hydrostatic pressure of the glomerular capillaries. So we get a net filtration pressure that pushes fluid out of the glomerular capillaries and into the capsular space. So since we compare net filtration pressure to glomerular filtration pressure, or rate, we want to basically acknowledge this term. This term, glomerular filtration rate, is the volume of filtrate, so the volume of fluid that is formed by filtering stuff through the filtration membrane, that amount and how much is formed per minute. So volume of filtrate formed per minute by both of the kidneys. So not just one glomerulus, but all the glomeruli in both kidneys. So it's actually about 120, 125 milliliters per minute. It's quite a lot of fluid. And the glomerular filtration rate is going to be proportional to a couple of things. One, we just talked about, net filtration pressure. And we know that whatever happens to net filtration pressure, same thing happens to glomerular filtration rate. But we also have to take into account the total surface area available for filtration. So if something's wrong with the filtration membrane, it could affect glomerular filtration rate and the filtration membrane's permeability. So if for some reason it's more permeability, hopefully it's not damaged, but that could definitely be something that causes it to be more permeable. That can change GFR. We really want a constant GFR. It allows the kidneys to make filtrate. It allows stuff to move through the tubular system at a good rate so that we can reclaim what we want. It isn't moving too fast. We don't miss reclaiming stuff to the blood. And so there's intrinsic controls which really maintain GFR, or glomerular filtration rate, for the kidneys, where they're going to make sure that the rate of filtrate formation stays about the same. The reason we want to do this is because if we see an increase in glomerular filtration rate, we see an increase in urine output. So an increase in the amount of filtrate made per minute means there's more stuff in the tube, but less stuff is being able to be reclaimed by the blood because the filtration rate is fast, so the stuff leaves the kidneys quickly. And that means if stuff's not in the blood, we've lowered blood pressure, and the vice versa will happen as well. So intrinsic controls will try to keep GFR pretty stable or pretty constant. But we do have extrinsic controls, which could negatively affect GFR, because extrinsic controls are trying to maintain systemic blood pressure in case of a hemorrhage or something else that's causing a drop in blood pressure. And so this could negatively affect the kidneys because overall systemic blood pressure is more important than blood pressure to one single organ. So the intrinsic controls use renal autoregulation. So we're going to see some dilation or constriction of arterioles. That's actually going to be the myogenic mechanism. And this is going to act locally, so it's acting only within the kidneys. And it's really just trying to maintain blood pressure in the kidneys just themselves. It's not looking at overall body blood pressure. The extrinsic controls, therefore, are not local. Instead, they're going to involve the nervous and endocrine systems. They're going to use mechanisms that maintain blood pressure for the entire body or systemically. And again, that can negatively affect the kidney function if we're not concerned, or the extrinsic controls are not concerned about the kidneys maintaining their GFR. But extrinsically, the systemic system, so the body as a whole, does take precedence over those intrinsic controls for maintaining GFR in the kidneys, especially if blood pressure drops below 80 millimeters of mercury or goes over 180 millimeters of mercury. So we're going to control glomerular filtration via glomerular hydrostatic pressure. So that's what we're going to be looking at, kind of to control or decide what to do about glomerular filtration rate. So if GFR, or for example, if hydrostatic pressure rises, it's going to cause NFP, or net filtration pressure, to rise, and that will affect glomerular filtration rate. So an increase in hydrostatic pressure increases the amount of pressure that pushes fluid out of the glomerulus into the capsular space, so therefore that's also going to affect the filtration rate of fluid moving through the nephron. And we see the opposite occur if hydrostatic pressure falls. But if it falls only 18%, so that's not much at all, GFR will immediately go to zero. So that's why it's really important for us to intrinsically control glomerular filtration rate so that glomerular hydrostatic pressure is not negatively affected. So the intrinsic controls are trying to maintain constant GFR, regardless of what's happening to mean arterial pressure, which is what's happening in the rest of the body. So mean arterial pressure, if you don't remember, is cardiac output with peripheral resistance taken into account that's occurring in the blood vessels. So outside of the range of 80 to 180 millimeters of mercury, when the extrinsic controls will take over, we're looking at GFR being maintained regardless of what happens to MAP. So there's two types of renal autoregulation for maintaining a nearly constant GFR in the kidneys, and that is the myogenic mechanism and the tubuloglomerular feedback mechanism. So myogenic, first thing you should notice about that word is myo. Myo is the prefix for muscle, so it has something to do with muscle, and it has something to do with muscle in the arterioles of our vascular system that's going to the glomerulus, and it's smooth muscle. So we're going to see that smooth muscle is responding when it's contracted or when it's stretched, and it responds differently if it's not contracted or stretched. So we're using mechanoreceptors here. So here's an example. If there is an increase in blood pressure, so an increase in pressure inside the blood vessels that feed the kidney, that causes stretch of the afferent arteriole. When the afferent arterioles mechanoreceptors are stretched, it causes vasoconstriction of the afferent arteriole, which will restrict blood flow to the glomerulus. If we restrict blood flow to the glomerulus, we are going to be reducing net filtration pressure and we'll be regulating glomerular filtration pressure. But this is a response to the original increase in blood pressure. Now, we're not going to be reducing GFR, we're going to be preventing a change in GFR because of that increased blood pressure. We're trying to respond kind of retroactively to an increase or decrease in blood pressure. We see the opposite occur with a decrease in blood pressure. So the dilation of afferent arterioles will occur if there's no stretch. Therefore, no constriction or vasoconstriction of the afferent arteriole. It'll dilate instead, and that therefore will allow more flow into the glomerulus. And if we need that in order to compensate for decreased blood pressure, that is going to maintain normal GFR despite any fluctuations we're seeing in MAP that are affecting kidney blood pressure. Next, we're going to look at the other mechanism for intrinsic control of GFR, and that is the tubuloglomerular feedback mechanism. Long name for a pretty simple mechanism. So this is a flow-dependent mechanism. So we're looking at filtrate flow, or the stuff that's in the filtrate going through the renal tubule system. And one of the things that's really in the filtrate is sodium. And sodium likes to combine with chloride, so we get sodium chloride concentration in the filtrate. Now, if GFR increases, so if glomerular filtration rate increases, meaning filtrate flow rate increases, or filtrate is moving through the tube faster, then we're not going to be able, when I say we're not going to be able, I really mean the tube is not going to be able to look at the filtrate and decide selectively what it needs to reabsorb. So it has less time to reclaim all of the nutrients and solutes and things that it wants back, that it wants to send back to the blood. So increased GFR speeds up how fast filtrate goes through the tube, which decreases the time for reabsorption, which means things get left behind. They stay in the tube. They don't go back into the blood. So that can actually lead to a lot of sodium and chloride, or sodium chloride, in the filtrate. And these high filtrate sodium chloride levels are going to be able to cause constriction or vasoconstriction of the afferent arteriole. And that constriction of the afferent arteriole is generally going to be occurring because of the release of renin, or the response by mechanoreceptors that are using those extraglomerular cells of the JGC to communicate between the afferent arteriole and the distal convoluted tubule. But it'll cause constriction of the afferent arteriole, which will slow down flow into the glomerulus, which decreases net filtration pressure. And we know if net filtration pressure decreases, so does glomerular filtration rate. And slowing down GFR was our goal, because originally we had our first stimulus was an increase. We wanted to slow it down, so we actually had time for that sodium chloride reabsorption. We want to reclaim the sodium and chloride that the body needs. And so if the opposite occurs, if GFR initially, as our stimulus lowers, we'll see the opposite. We'll see vasodilation of the afferent arteriole, we'll see NFP and GFR increase, and we'll have less time for sodium chloride reabsorption. But these things aren't drastically changing from not enough time to a lot of time, not enough time to a lot of time of reabsorption. We're not constantly trying to change GFR. These mechanisms are slightly changing GFR or NFP in order to keep a constant GFR, glomerular filtration rate. So they're not going to cause the blood pressure or the GFR, the NFP to bounce, unless we were using extrinsic mechanisms, because then we're not really concerned with what the kidney's doing. We're trying to kind of save the human body. We're trying to maintain mean arterial pressure in that extent. So again, under normal conditions at rest, it's going to be the intrinsic controls that dominate. The renal blood vessels will be dilated, renal autoregulation mechanisms, they're prevailing. But if it's not normal conditions, if we're outside of that 80 to 180 millimeters of mercury blood pressure for mean arterial pressure, then extrinsic controls are going to take over. And these are mainly going to be regulated by the sympathetic nervous system. So here's an example. If extracellular fluid volume is extremely low, so blood pressure is low, then norepinephrine or epinephrine will be released by the nervous system via the adrenal medulla. They're released to the blood, so they're sort of acting as hormones, but the adrenal medulla is really close to the kidney, so it helps us kind of visualize where this is occurring. But the release of norepinephrine and epinephrine into the blood causes systemic vasoconstriction. So vasoconstriction will put more pressure on the blood inside blood vessels, therefore increasing blood pressure or increasing peripheral resistance, which increases blood pressure, and that will occur in the entire systemic circuit. Now, constriction of afferent arterioles to lower GFR may occur as a result of these extrinsic controls, but we may not be able to respond quick enough, and this could increase blood volume and pressure as a result in the kidneys minutely before GFR is able to go back to a constant rate. But the extrinsic controls, again, are not concerned with the kidney. They're trying to maintain kind of body-wide blood pressure so that all other organs are able to get the fluid and the nutrients and the oxygen they need. So if you need to review the main mechanisms for increasing blood pressure, such as the autoregulation that involves not only the renin-angiotensin-aldosterone mechanism, but also autoregulation by changing the diameter of arterioles, go back to chapter 15 and 18. But the three pathways that you'll probably see that are for renin release that are applying here to the extrinsic controls and the intrinsic controls of renal regulation is the direct stimulation of granular cells. So the sympathetic nervous system can directly stimulate the granular cells, which secrete renin, which can cause vasoconstriction. The stimulation of activated macula densa cells when filtrate sodium chloride concentration is low. So the macula densa cells are monitoring that sodium chloride concentration, and they can cause renin release by talking via the extra glomerular mesangial cells to the granular cells and telling them to release renin. But they want to either lower or increase filtrate flow depending on what's happening to GFR. And then also reduced stretch of granular cells can cause them to release renin, which can cause vasoconstriction or vasodilate. All right, we're finishing up. So extrinsic controls. Other factors we want to talk about affecting GFR are adenosine, prostaglandin, intrinsic angiotensin 2, which is going to reinforce the renin-angiotensin-aldosterone mechanism. All of these things are chemicals that, if released, can act as paracrines and affect those afferent arterioles. And that is it for Part A on the urinary system. Make sure to go back and review the anatomy and then add that physiology to that, and just make sure you're comfortable with the structure and function of this part that we've talked about for the urinary system.